In the previous video, we have just touched upon how this T cell development occurs in the thymus. In this video, we will cover in brief how this T cell development occurs in the thymus. So, these T cell progenitor cells usually grow in the bone marrow in the case of adults, and from there, based on their cues, they will be directed towards this thymus organ. And when they reach the thymus organ, they don't have these CD4 plus and CD8 plus receptors, and those progenitor cells that those progenitor T cells that does not have the CD4 plus and CD8 plus receptors are being termed as these double negative receptors. And these double negative receptors has this characteristics, that is they have CD44 plus receptors, however they don't have these CD25 receptors. After that there is a rearrangement occurs, and when till they reach to this DN3 stage. In the DN2 stage, they have both CD44 plus and CD25 plus receptors. However, when they reach the DN3 stage, they don't have these CD44 receptors, whereas they have these CD25 receptors, and this is in completely reverse that of DN1 condition that we can see here. In the case of DN1, they have CD44 plus and CD25 minus. That means they have CD44 receptors and does not have CD25 receptors. Whereas in the case of DN3, they have CD44. They don't have CD44 receptors, whereas they have CD25 receptors. And after that, when they reach at this DN3 stage, this rearrangement of the beta chain occurs, or TCR, T cell receptors beta chain rearrangement occurs, and this beta chain rearrangement will leads to the formation of this immature single positive. Immature single positive means that the rearrangement is still not completed, and they might have this CD4 plus or CD8 plus receptors during this rearrangement process. And when they reach this immature single positive condition, they further proceed for this TCR alpha rearrangement, and this TCR alpha rearrangement will make them the mature double positive T cells that have both CD4 and CD8 plus T cells. And finally the thymus produces the double negative T cells into this double positive C cells that have both CD4 plus and CD8 plus receptors. One more thing that we need to understand is that when this TCR beta rearrangement going on, there is also possibility that there is that TCR delta or gamma rearrangement might also occurs at small percentage, and those cells will not further proceed towards this TCR alpha rearrangement, whereas those delta TCR delta cells will finally proceed towards apoptosis, as they will not be able to survive since within this thymus, as they don't acquire the CD4 and CD8 plus receptors, or in other terms they do not have the proper rearrangement of these T cell receptors. The cells till from this DN1, DN2, DN3 and DN4 stage are being termed as double negative, as we can see here, and the number also represent the different stages of the double negative that I have, or that can be easily understood by the receptors that are present and absent from the figures. So this is just a general description of how this T cells development occurs in the case of thymus. The cells, the homeopathic stem cells from the bone marrow, they first reaches to the thymus, and this after this whole rearrangement, the homeopathic stem cells from the bone marrow, uh, they first these stem cells have the capability to form into any other cells. So these homeopathic stem cells first form this early thymus early thymus committed lineage, and after that they will move towards this thymus based on the cues that are present on the this early progenitor thymus cells, and thereafter when they reach the thymus the process will occur and the T cells will finally, the T cells that leaves the thymus will have this both CD4 plus and CD8 plus receptors. So if you need to understand more about this T cell development in thymus, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.